My name is Barbara Wendelberger, and I'm a biostatistician at Berry Consultants. Today I'm going to be discussing the importance of dose selection in clinical development. The clinical development timeline is comprised of 5 major phases. Extensive preclinical research is used to determine which promising therapies will be tested for safety and efficacy in later stages of clinical development. Dose selection occurs while investigating safety in Phase 1 and while exploring efficacy in Phase 2. Phase 1 and Phase 2 trials tend to have smaller sample sizes and to dedicate fewer resources to dose selection. This means that there is a higher probability of selecting a suboptimal dose to be carried forward into the next phase. Selecting a dose that is too high may result in adverse events that prevent further clinical development, while selecting a dose that is too low may result in subtherapeutic effects and failed clinical trials. There is an accumulating effect of the decisions made during the clinical development process. For example, there is no power to detect the efficacy of an uninvestigated dose. Thus, putting all of your eggs in one basket, meaning that you only investigate one dose, increases the risk of failed drug development. There are many reasons that clinical trials fail. Failure in Phase 3 may indicate ineffective decision-making earlier in clinical development. One example is Alzheimer's disease. In a 2014 Cleveland Clinic study, the authors found that over 98% of Alzheimer's treatments failed in Phase 3 over the time period studied. Could some of this be due to poor dose selection in early development phases? In addition to failed trials, delays due to scientific and regulatory reasons may also stymie clinical development. In this publication, authors at the U.S. Food and Drug Administration focus on new drug developments. Of the 151 unsuccessful first-time applications, 15.9% included uncertainties related to dose selection. The authors went on to conclude that several potentially preventable deficiencies, including failure to select optimal drug doses, accounted for significant delays in the approval of new drugs. Incorrect dose selection can also have post-marketing implications. In this study of 499 FDA-approved new molecular entities from 1980 to 1999, dosages of 1 in 5 of those new molecular entities was changed, underscoring the need to improve pre-marketing optimization of dosage and indicated population. We will now talk about some strategies to improve dose selection. Our first strategy is to increase sample size. This can be done by running larger Phase 2 trials and also by using seamless Phase 2-3 trial designs. Our second strategy is to increase the number of dose levels being investigated. This can be done through dose response modeling and also through response adaptive randomization, or RAR. So the question is, can we innovatively and efficiently implement these strategies? There are many reasons to increase sample size. The first reason is to obtain a better estimate of the treatment effect. This is an issue of particular relevance when we are trying to estimate differences in effect between different dose levels. The second reason is to increase power, which reduces the likelihood of false negative findings. There are still some real-world limitations, which include patient resources, financial funds, and clinical trial infrastructure. So while this all sounds good theoretically, you're likely thinking, I don't have the resources to increase sample size. Here are a few strategies to make this more feasible. In a seamless Phase 2-3 trial, Phase 2 information can be leveraged into Phase 3. Dose response modeling can borrow information across doses, meaning that increasing the number of doses does not correspond to a linear increase in the sample size. Response adaptive randomization is a patient-centered and statistically efficient method for dose selection. In a seamless Phase 2-3 trial, we can thoroughly explore efficacy before moving on to the confirmatory portion of the trial, and we can leverage resources from our Phase 2 into our Phase 3. This may include evidence-based dose selection, shorter development time, and an efficient use of patient and clinical infrastructure resources. Using seamless Phase 2-3 trials, we can efficiently investigate multiple doses in Phase 2 to inform Phase 3. We can explore a larger dose grid using dose response modeling. This approach allows us to learn about the relationship of the response across a range of doses, to gain important insight into how the response may vary as we increase or decrease the dose. Dose response modeling includes information from all patients at all doses to inform estimation of the treatment effect. The dose-response relationship can be used to select the optimal dose for subsequent trials. Response adaptive randomization, or RAR, preferentially randomizes patients to better performing treatment arms, which allows us to drop ineffective arms from the trial and to increase the probability of better patient outcomes. With more patients allocated to better performing doses, RAR improves statistical efficiency for estimation of the treatment effect at the most relevant dose or doses. So RAR provides better patient care while increasing statistical efficiency. Now we will walk through a real-world example. Dulaglutide was investigated as a treatment for diabetes in an adaptive dose-finding seamless phase 2-3 clinical trial. A predefined adaptive sample size ranged from 200 to 400 patients in the phase 2 portion of the trial and included 7 doses of the active investigational agent as well as a placebo and an active comparator. Patients randomized in the phase 2 portion of the trial were randomized using response adaptive randomization. Part 1 explores the efficacy. This is the phase 2 portion of our trial. Patients are randomized to one of 7 doses, a placebo, or an active comparator. Interim analyses occur following a burn-in period of 50 patients who are randomized equally. RAR begins with the interims and occurs throughout Part 1. Beginning at 200 patients and ranging up to 400 patients, the trial could stop for futility or could shift from Part 1 into Part 2. If the trial goes up to 400 patients, a decision is forced at that 400 point. The phase 3 portion of this trial is powered by the phase 2 portion. Everything here is prospectively defined, and this includes the algorithms, the rules, the decisions, and the analyses. Part 2 of the trial is our confirmatory phase 3 portion and investigates the 1 to 2 doses that are selected from Part 1. Patients are randomized to the selected dose or doses, placebo, or the active comparator. Patients are leveraged from Part 1 of the trial, meaning that all active patients from the selected doses are compared to all control patients, including those from Part 1. So what happened when this trial ran? The trial shifted from Part 1 to Part 2 at 200 patients, which was the minimum predefined sample size at which this shift could occur. In Part 1, 2 doses were selected to test in Part 2. The entire adaptive trial was prospectively defined and ran exactly as planned, spawning other phase 3 trials. Ultimately, the FDA approved dulaglutide as a treatment for diabetes, with a peak sales projection of $1.3 billion in 2020. So what is the takeaway message? Effective dose selection early on in clinical development has substantial downstream ramifications. Increasing sample size and resources devoted to early phase development improves dose selection. Feasible strategies to accomplish this include seamless phase 2-3 trial designs, dose response modeling, and response adaptive randomization. Correctly identifying appropriate dosage has scientific, regulatory, and healthcare impacts. Thank you. I hope you learned something about dose selection.